Assalamualaikum and hi. So today our group will be presenting you guys article about antibacterial activity of silver nanoparticles confined to mesostructure silica-based calcium phosphate against methicillin-resistant Staphylococcus aureus, which is MRSA. This is the reviewers of this article. There are two major discoveries based on this article. Firstly, silver nanoparticles effectively inhibit bacteria and virus due to their strong reactivity influenced by particle size, like silica-based calcium phosphate with 3% of silver was synthesized exhibiting antibacterial effect against E. coli, SRS and Pseudomonas aeruginosa. Next, the researcher hypothesized that mesostructure silica-based calcium phosphate can effectively inhibit MRSA while providing bioactivity that enhancing bone graft properties. There are four objectives have been discussed in this article. Firstly, to synthesize mesostructure silver-containing silica-based calcium phosphate with bioactivity and antibacterial properties to determine the minimum inhibitory concentration MIC of silver 80 against MRSA, to evaluate the in vitro bioactivity of silver 80, the carrier virus susceptibility test, the kinetic of microbial crew, and to analyze the relationship between silver 80 antibacterial activity and composition, time, and dosage.
In this slide, I will explain about three major methods in this research. The first method is in vitro bioactivity testing. The sample, which are 80% more silica-based calcium phosphate, 1% and 10% silver with silica-based calcium phosphate, were immersed in the phosphate buffer saline PBS to assess their bioactivity over different time intervals using scanning electron microscopy. The purpose to evaluate the formation of the bioactivity of silica-based calcium phosphate sample on the sample surface over different time interval, which are 10, 24, 48, 72, and 168 hours. Next method is the characterization of silica-based calcium phosphate powder. The silica-based calcium phosphate powder were characterized by adding them to a soy broth, oscillating and filtration, then analyzed using the UV absorption spectra and transmission electron microscopy (TEM). The purpose of the method is to obtain a colloid and to assess particle size and dispersion of the sample. This method is disk diffusion analysis, or known as a Cabri barrel test. 0.05 gram of different silica-based calcium phosphate sample were pressed into a disc and placing on a agar plate that inoculated with S. aureus and MRSA. Purpose is to assess the antibacterial effect of the different silica-based calcium phosphate, S. aureus and MRSA by measuring the inhibition zone size around powder disc. Result of major method. In vitro bioactivity testing, the results show the formation of an apatite-like layer on the surface of the sample, indicating their potential for the bonding to living tissue, such as bone tissue. Next, result of the method characterization of silver with silica-based calcium phosphate powder. For the UV absorption spectra, the 1% silver with silica-based calcium phosphate sample has a maximum absorption peak at 4 to 8 nanometer, and the sample of 10% silver with silica-based calcium phosphate were at 306 nanometer, which confirm the presence of the silver nanoparticle. For the transmission electron microscopy, the 1% silver with silica-based calcium phosphate samples has average particle size about 5 nanometer and uniform particle size distribution, while the 10% silver with the silica-based calcium phosphate sample particle size are 5 nanometer to 200 nanometer, and the particle size distribution is not uniform. The result of the disk diffusion analysis method, 1% and 10% silver with silica-based calcium phosphate sample exhibited antibacterial effect against S. aureus and MRSA by forming inhibition zones around the powder disc. The diameter of the inhibition zones arranged from the 16mm to 90mm, indicating the antibacterial activity of the samples.
Next, I proceed to the next part, which is, can any of the results be interpreted differently? The answer is no. This is because the study demonstrates in vitro bioactivity and antibacterial evolution of Ag per ATS bioactive ceramics powders against S. aureus and MRS were identified. The results and data were presented in tables and supported by figures. The statistical analysis reinforced the study conclusions. The author state and discussed the result in the context of easy to understanding the in vitro bioactivity and antibacterial evolution of Ag per ATS bioactive ceramics powders against S. aureus and MRSA. In conclusion, the in vitro bioactive and antibacterial evolution of Ag per ATS bioactive ceramic powder against S. aureus and MRSA was studied. Ag1 per ATS can form an epithet-like layer on the surface after 48 hours in PBS. SRD pattern of Ag10 per ATS showed on lipids for silver and silver chloride. The analysis of microbial growth kinetics and the colony forming capacity assay confirmed that the Ag1 per ATS powder have antibacterial activity against S. aureus. The minimum bactericidal concentration was not determined in any case.
Yes, I agree with the conclusions of the author regarding the antibacterial activities of silver nanoparticles against methicillin-resistant Staphylococcus aureus (MRSA). The study provides comprehensive evidence throughout various experiments, including turbidity measurements, colony forming capacity assay, and disk diffusion method, to demonstrate the inhibitor effects of silver nanoparticles on MRSA group. The researcher effectively utilized different analytical techniques to assess the minimum inhibitory concentration (MIC) and minimum bacterial concentration (MBC) of the silver nanoparticles, providing a thorough understanding of their antimicrobial properties. The author did not mention or considering conflicts or supporting evidence from other studies in their research. However, in these fields of antibacterial activity of single nanoparticles, there are various reports with both supporting and conflicting evidence regarding the effectiveness of single nanoparticle against antibiotic-resistant bacteria like MRSA. Factors such as nanoparticle characteristic and experiment condition can influence the outcome. Discussing this diverse finding would have provided a more comprehensive view on the topic. One limitation of the research is the lack of discussion on the potential cytotoxicity or adverse effects of single nanoparticles on human cells of tissue. While the study focused on the antibiotic activity of nanoparticles against MRSA, it is essential to consider safety profile of this nanoparticle for potential clinical applications. Furthermore, the research could be strengthened by including in vivo study to validate the findings observed in vitro, while the in vitro experiment provide valuable preliminary data. Translating this result to animal models or clinical trial would offer a more comprehensive assessment of the potential therapeutic benefits of single nanoparticle in treating MRSA infection. In vivo study would provide insight into nanoparticle pharmacokinetic, biodistributions, and overall efficiency in a more complex biological environment.